So when we are studying the biotechnology, the vaccination also becomes an important part in this. So we will know about the vaccination and the 4 generations of vaccines, right? Good morning, students. Welcome back to Plutus IS. So, right, today is our 30th day, right? So, till now we have completed 29 topics, right? So, one more important update is also there. So, because of the general elections, the prelims examination has been, we can say, postponed to I think 16th July. Sorry, 16th June. Earlier it was scheduled on 26th May. So you will get like 20 days more for the preparation of prelims exam. So I try to utilize this extra period and modify your whatever the plans have been made. So your timetable, etc. So try to modify them also. So because we have got some extra time, I will try to cover. Till now the plan was to cover 95 topics. So I will try to cover, add 5 more topics, and I'll try to make the topics as 100. Let's try and see how far it will be possible, right? So the major point is try to utilize the extra time, whatever the 20 days you are getting, try to properly utilize them, right? Right, today the topic for discussion is biotechnology. As you all know, this is also an important area when it comes to the prelims examination. In the past also we can see lot of questions. Many questions were asked from the GMO, genetically modified organisms. Similarly, biotechnology. It is a broader area. So subtopics like gene therapy is there, and artificial production of vitamins, vaccines. So ever since the COVID incident, COVID has occurred, the about the questions about vaccines, so they are frequently being asked. Also the generations of vaccines, there are 4 generations of vaccines, so questions are being asked from those areas also. So try to focus on this topic also. It is very, very important from the point of view of examination, right? So biotechnology, if you see the definition, so it is derived from the words bios, which means biology, and and technology, it's referring to industrial application of living organisms and biological process. So basically the biotechnology, it is derived from the words bios, and technology has been added to it. So biotechnology refers industrial application of living organisms and biological processes. So try to remember the definition, right? So some of the key processes that are involved in the biotechnology are, it encompasses biochemistry, microbiology. Microbiology is studying about the microorganisms. Like another important we can say area that is related with the biotechnology is genetic engineering, right? If we see briefly, we see the applications of biotechnology, healthcare and medicine. So fighting infectious disease, biotechnology is helpful in fighting the infectious disease such as SARS and influenza. So these are very dangerous diseases. We have seen bird flu kind of diseases, and the latest example is COVID-19. And we have seen the adverse impacts of these diseases. So biotechnology helps fight these diseases. The best example is we can develop vaccines, and when people are vaccinated, so people get, the populations get, we can say, immunity towards these, we can say, viruses, right? So it also helps in the development of more effective pharmaceuticals in fighting these, whatever the infectious diseases are there, right? Similarly, another advantage is development of vaccines and antibiotics. So microorganisms such as Bacillus, Polymycia and Penicillium notatum, they are utilized to produce antibiotics like polymyxin, B and penicillin. Penicillin, as you all know, it is one of the first vaccines that have been discovered or invented. So these help in fighting various kinds of diseases, we can say, right? Similarly, another important application is treating genetic disorders. So genetic disorders are there, like leukemia is there. So these kind of genetic disorders, it is very difficult or it is impossible to fight these diseases with the normal medicines. So biotechnology help us treat these genetic disorders, right? So gene therapy, through the process of gene therapy, it holds the promise of replacing defective genes because of which the genetic disorders are occurring. So we can through the gene therapy we can replace the defective genes with the normal ones to treat the diseases like inherited disorders. For example, diabetes is the best example. So diabetes, it is, some of the major proportion of the diabetic, it is genetic also. So these kind of disorders can be fought. Similarly, it is applicable in forensic science also. So techniques like DNA fingerprinting, and they aid in finding out the criminals and to solve the criminal cases, right? If we see the environmental applications, cleaning up and managing environment. So generally the environment has it has been polluted with heavy metals also. When we were studying the pollution, we have seen. So mercury like pollutants, when they enter environment, they act as pollutants, and they cause severe health impacts on the human beings and on other animals. So through the process of bioremediation, we can bioremediation involves using living organisms, including genetically modified organisms, to break substances into the environment. So basically what happens, bioremediation in this, so these agents will be we can say involved in the area, cleaning of an area. For example, so a land area is affected by mercury pollution or some other heavy metals. So bioremediation to another sub branch is phytoremediation. Right. So phytoremediation, particular type of plant will be grown in that area. So that particular plant will absorb the we can say the hazardous we can say particles or the heavy metals, and in that way it will clean that area. So phytoremediation. Similarly, in bioremediation, for example, oceans are polluted by the oil spills, and it is called as oil pollution. So one particular microorganism is there, it is genetically produced, that is oil zapper, right? So when this organism is, we can say it is released there, it will consume the oil particles and it will clean the area, right? These are the examples of bioremediation. So these organisms, the oil zapper, or for example any kind of bioorganism, microorganism, or even the plants which are used in phytoremediation, they are produced through the we can say techniques of biotechnology, right? If we see the use of biotechnology in agriculture, biotechnology helps in improved agriculture or crop production, right? Biotechnology enables the development of disease resistant, herbicide resistant, and insecticide resistant crop varieties. So through which we can realize more and more production, right? Similarly, it will help in pest control, right? So genetic manipulation of pests can be render them sterile, controlling their population effectively. So genetically modified, we can say, genes can be inserted into the pests themselves, so that the pest will become sterile. Sterile means they cannot produce their offspring further. So in this way they are being eliminated, right? If we see the applications in industry, manufacturing and bioprocessing. So biotechnology helps in manufacturing and bioprocessing of products. So biological techniques facilitate large scale production of plants, yielding compounds for various industrial applications such as detergents, paints, lubricants, and plastics. So in all this production of all these products, biotechnology techniques are used or utilized. Next, food and beverage processing. So biotechnology simplifies food processing and preservation, leading to development of seedless fruits and improved food storage methods. So seedless fruits, best examples are grapes. So earlier grapes had seeds. So through the biotechnological methods, we are able to produce now seedless grapes. Similarly, in fruits like papaya also, we will see less number of seeds. Earlier there used to be many seeds inside the papaya fruit. Now we can see the fruit with very less number of seeds. So it is this is made possible through the biotechnology. Similarly, through the biotechnology we can also invent better storage mechanisms, right? Similarly, we can develop new products also. So biotechnology is utilized in the industry to develop food additives and aid in the mass production of items like cheese, yogurt, and alcohol. So in these processes, mass production of these products, the fermentation process is utilized, right? So through the fermentation process, mass production of these consumable goods, food products, they are being produced mass quantities, right? So these are some of the applications of the biotechnology. Try to remember them. Not only in prelims, they are also useful in mains examination also, right? Now we will see industrial microorganisms and their products. So industrial microorganisms basically mean they are generally produced through the biotechnology only. They are produced through biotechnology only, right? First one is yeast. Yeast, it is basically comes under fungi, classified as a fungi. So it is important for fermentation process in industries. So fermentation is used in many processes like producing alcohol beverages. Even it is produced in bread production. It is used in bread production also. In baking of bread also, the yeasts are used. Next is molds. It is also one kind of fungi. It is used in various industrial processes including fermentation, enzymes production. So try to remember this part also. So molds are used in fermentation and enzyme production. Similarly, bacteria, right? It is widely used in industrial biotechnology for various purposes. We will understand in what purposes bacteria used, right? Next is filamentous bacteria. So it is valuable in industries for their role in producing antibiotics and other bioactive compounds, right? If you see the products manufactured using microorganisms, the products are alcohol containing beverages. So in this, yeasts are used for fermentation in brewing and wine making. Next, yogurt. It is one form of curd, right? So certain bacterial strains are used in yogurt fermentation. Proteins, microorganisms are engineered to produce specific proteins for industrial purposes. Next, antibiotics and monoclonal antibodies. These are used in bacteria and other microorganisms are employed in the production of antibiotics and monoclonal antibiotics. Next, the production of vitamins, steroids, and enzymes. Right. Microorganisms are harnessed to synthesize vitamins, steroids, and enzymes used in various industries. Similarly, in production of biogas also, microbial fermentation is utilized in biogas production as a renewable energy source. As you all know, biogas is a renewable energy source. Right. So here also microbial fermentation used as a technique in the biogas production. Right. So when we are studying the biotechnology, the vaccination also becomes an important part in this. So we will know about the vaccination and the 4 generations of vaccines, right? So basically, origin and development if we see. So in 1796, Edward Jenner, he observed that milkmaids exposed to cowpox were immune to smallpox, right? He observed that, and he conducted an experiment by infecting a boy with cowpox, who later showed immunity to smallpox, right? So he coined the term vaccine. So it came from the Latin vacca, which meaning cow, for the weakened germ and its protective inoculation, right? So principle of vaccination, if you understand, he proposed that introducing attenuated or weakened germs into the body would provide immunity without causing disease. So this is the formula behind the principle of vaccination. So when we introduce weakened, or for now, for example, we are also using the dead, we can say dead germs also, or microbes also in vaccines. So the principle behind the vaccination is, when weakened, we can say germs are introduced into the human body, so they will, instead of infecting the human being, they provide immunity to the human being. So best example is in polio vaccines, we are using the dead or the live attenuated microorganisms. When they are injected into the children, so they slightly produce the response of the immune system. So next time when actual polio virus attacks human body, so the body's immune system will be in a position to react and produce antibodies to fight that, we can say, polio virus, right? Now we will see the evolution of vaccines. So first generation vaccines, they are made from attenuated disease causing bacteria. So this technique also used by Edward Jenner. Next, examples include vaccines for smallpox, measles and mumps. Second generation vaccines, they are produced through genetic engineering or recombinant DNA technology, right? We will later understand what is exactly meant by recombinant DNA technology. Some of the examples are vaccines for hepatitis B virus and herpes virus. Similarly, third generation of vaccines, they are synthesized from chemicals. So remember this one, they are actually synthesized from chemicals, right? So examples include some influenza vaccines. So we have seen H1N1 is there, and H1H6 is also there, H1N6. So there are many influenza viruses nowadays. So some of the examples are vaccines for these influenza viruses, right? Now the fourth generation of vaccines are also there. They here a biochemical molecule constructed artificially for the purpose of vaccination. So here not a complete organism is there, only biomolecule is there. Molecule is there. It is achieved through the process of, we can say, biotechnology, employing the biotechnology. So the latest example is some of the vaccines produced by Pfizer. So the mRNA vaccines, it is the example of fourth generation of vaccines, right? So try to remember this these aspects, generations. So there may be a question about matching. So here we can say in part first column, there can be like generations, first, second, etc. Third, fourth. Here there may be a pairing of example or the technology involved. So there can be a matching, and the question be like, which of the below pairs are correctly matched or incorrectly matched? Then you have to find out the right option, Right. So these kind of questions are possible. So try to remember these 4 generations of vaccines. Right. So if we understand the present example of vaccines, so it has become cornerstone of public health, preventing the spread of numerous diseases. So especially in developing countries like India, so vaccines are playing an important role. So because we do not have proper health facilities, the health services are not reaching, especially to the hinterland areas. So to prevent, in this case, to prevent diseases, vaccines have become very, very important. Vaccines have become importantly. So for that matter, we can say we are running the largest vaccination program in all over the world. So you might be heard about the Indradhanush program. Indradhanush program. So here a combination of vaccines are given to children to protect them from various diseases. Similarly, we have seen the COVID and its havoc. So in the to fight the COVID-19 also, the vaccinations have played an important role. Right. So especially in developing countries like India, the vaccination and their utilization to fight the infections. So it is very, very important from the health perspective, especially in the developing countries like India. Right. Now we will see another important aspect, production of vitamins using biotechnology. As you all know, vitamins are very, very important. Apart from the proteins and carbohydrates, vitamins also plays an important role in the human body, or we can say human health. So these are macronutrients, and especially the vitamins are known as micronutrients. So whenever the vitamins are, we can say, not enough in a human body, we call that as hidden hunger. So basically, the present day children in the present day, the children are facing children of rural India facing this particular problem. Though they are taking sufficient quantity of food, the nutrients, micronutrients, especially vitamins, are lacking in their food. So because of this, they are falling prey to various kinds of diseases. So to address this, one program that has been initiated is, we have seen the the millets have been are being promoted, we can say, actively in the consumption of the in the food consumption, especially to the children of rural areas. So they have a lot of, we can say, micronutrients. Right. So this is one way we can say. But earlier, to fight the lack of nutrients and to give vitamins, or to give the vitamins in sufficient quantities to prevent the diseases, so we also employed biotechnology to produce them in sufficient quantities and to give to the people who are in need of them. Right. We will understand that process also. Right. So introduction to vitamins. They are essential nutrients required in small amounts for various metabolic reactions in the body. So they play crucial role in maintaining health and preventing deficiency diseases. Right. If you see the historical perspectives, so vitamin C, it is the first vitamin to be produced through the process of biotechnology. So it is produced through a fermentation process utilizing bacteria. Similarly, vitamin B12, it is also called as cyanocobalamin, and B2 riboflavin. So it is obtained from, earlier it is used to be obtained from liver extract. So when biotechnology process was there was, we had to kill lot of animals, like even the B12 has been adapted from the killing the we can say camels also. So basically it has been synthesized, or we can say produced from the liver extract. So liver, in this process the liver of the animals has been utilized. So production of, with the introduction of biotechnology, the production of vitamin B12 has been simplified, or we can say it was made more easy. So it is by using fermentation by propionic bacteria, the artificial synthesis of B12 has been made possible. Right. Similarly, some of the production of some of the other vitamins, it has been enhanced through the process of biotechnology. If we take the example of vitamin B12, riboflavin, so it is generally naturally found in cereals, vegetables and yeast. So microbes are used to enhance the yield of B2 significantly, right? So microbial fermentation can increase the yield of B12 100 to 300 fold compared to natural sources. So in this way also the production of B2 has been revolutionized. If we understand the significance of biological production of vitamins, so biotechnological methods have revolutionized the production of vitamins. They offer more efficient and cost-effective means of obtaining vitamins compared to traditional extraction methods. So imagine to obtain B12 cyanocobalamin, we have to kill animals. So now there is no need for killing animals. So we can artificially produce the B12. So in this way, the production of vitamins has been made a lot easier. So production of biogas also. So we have understood it is a renewable energy. It is more relevant in rural areas because dung basically used, that is the waste, animal waste, cow dung, or we can say dung from the animals is used. So it is basically, so it produces energy. One way it produces energy, and people were using this energy for cooking gas. So similarly, whatever the byproducts comes in the biogas production process, the slurry, it is a good manure. So right, so in this way the biogas production process, we can say proved very useful, especially in the rural areas. So basically biogas is a renewable energy source that can help save fuels like coal, kerosene and petrol, which are depleting rapidly. Right. The source and composition, if we understand, it is primarily made from organic waste, including fecal material of cattle, which contains lignocellulose. Right. Methane, CH4, it is the main component of biogas and it serves as the energy source. So people used to use this biogas in the cooking process. Remember this point, in this biogas, methane is the major gas present. Other gases are also present. So if you process understand, basically the major process involved here is fermentation, which comes under the biotechnology. Right. So produce a major components in this biogas we have understood. Methane is the major component. So some part is also containing carbon dioxide. Right. So basically through various chemical processes, series of chemical reactions involving fermentation, this biogas is being produced. Right. Next is another important area, genetic engineering. So there have been questions from this area also. So try to focus and understand this area also, genetic engineering. So it involves manipulation of genetic material such as DNA. We have understood chromosomes, DNA, to alter or replace genes and genetic products. So this is the definition of genetic engineering. It involves manipulation of genetic material such as DNA to alter or replace genes and gene products. Right. Conceptualization. So Genes, the nucleotide sequence of in DNA, they contain the information for functioning of the body, similar to instructions for a machine. So whatever the functioning of the human body, so all that information is stored in the gene level at the DNA. It is stored in the sequences of DNA, right? So progress in molecular biology has led to the development of techniques for manipulating the genetic material, making such as making identical copies of genes. So it is called as cloning, and similarly storing them in gene libraries so that it can be used in the future. If we understand the importance of genetic engineering, so it is used in curing several diseases. So for example, diabetes mellitus, it is basically diabetic diseases. So diabetic patients, they lack a gene responsible for insulin synthesis. So insulin is required for absorbing the glucose, whatever we get, whatever that is generated from the food we are eating, and the insulin plays a role in absorbing this glucose and releasing the glucose whenever it is required by the human body. So due to the we can say default in the gene, a particular gene, this insulin production is not happening in a particular human being, right? So through genetic engineering or gene therapy, we can correct this. We can say anomaly. So that through the newly introduced, we can say, genes, the insulin can be produced in the human bodies, right? Next is thalassemia. So it involves lack of gene. It is because of the lack of gene for hemoglobin production, requiring frequent blood transfusions for survival. Similarly, sickle cell anemia is there. So it has the people who have been affected by this disease, they have an altered gene resulting in abnormal red blood cells due to faulty hemoglobin. So in these people who have sickle cell anemia, so they have different structure of hemoglobin. So it becomes difficult for blood to transmit within the human body. So because of this, the organs or we can say flesh, they do not have the sufficient oxygen levels in the body. So because of this reason also they suffer, right? So application in genetic disorders. So genetic engineering offers hope for individuals who are suffering the diseases like this: diabetes mellitus, thalassemia and sickle cell anemia. It hopes offer for individual with genetic disorders by enabling the production and storage of genetically engineered DNA copies in gene libraries for future use, right? If we understand the industrial applications, so it is utilized in industries for production of various proteins on a commercial scale. So bacteria are genetically manipulated to serve as biological factors for producing enzymes, hormones and antibodies and also vaccines. Genetically engineered bacterial strains, they are developed to degrade harmful environmental pollutants. So just now we have seen bioremediation, right? And under this we have also seen the example of phytoremediation. So these are possible. So here producing enzymes, hormones we have seen. Similarly, vaccines also we have understood 4 generations of vaccines. So all these are possible through the biotechnology, especially through the technology of gene editing or gene therapy, right? Next, one of the important components of gene therapy is recombinant DNA technology, right? So this is the major components in the gene therapy, right? So definition, if we see, genetic engineering involves the construction and utilization of new DNA molecules engineered through recombinant DNA techniques. So recombinant DNA technology, it entails cutting a segment of original DNA. It involves cutting off a segment of original DNA and replacing it with a different segment of DNA contained desired characteristic. So if this is the DNA, generally the structure of the DNA will be like this. If we say this part of DNA, it is we can say malfunctioning, and because of this we can say, I'm explaining with an example. So if this part of the DNA structure, it is causing abnormality, for example sickle cell anemia, or we can say diabetes mellitus. So through the recombinant DNA technology, this part is removed, and a new part, let's say a new strain, a new DNA part is introduced here, which is, I mean, proper one, which is a correct functioning strain. So here a new we can say chromosome is produced, and this we can say works properly. If we say if we take the sickle cell anemia, so here this gene once it is modified, it will produce the proper red blood cells. Or if we see the diabetes mellitus, it will start producing the insulin, right? So it is the gene therapy. So especially the technology known as recombinant DNA technology. So whatever the defect we can say gene portion, a genetic portion, it is removed, and the properly functioning gene portion, it is we can say introduced in the chromosomes. So it is this is the technology involved in recombinant DNA technology, right? Key terminologies that is involved in recombinant DNA technology, if you understand. Recombinant DNA, so it is resulting from the insertion of a foreign DNA segment into plasmid or other vector. So I mean here the plasmids they act as a vector. I mean they break this malfunctioning we can say genetic portion, and it will introduce the new portion which is properly functioning, right? So it is it is known as recombinant DNA. Whatever the DNA introduced there, it is known as a recombinant DNA, right? Next, clone. It is a group of genetically identical cells, descendants for a single cell, that is known as clone. Cloning process, if you see, it is the production of genetically identical individuals or genetic material from a single cell. So cloning is a cell can be taken from my body, and another person identical to me can be produced. It is possible from the present available technology. It is known as cloning, right? If that happens in the plants, it is known as plant cloning, right? Right. If you understand the foundations of recombinant DNA technology, there plasmids play an important role, right? So here extra chromosomal DNA fragments in bacterial cells that replicate along with the bacterial DNA and serve as vectors for carrying foreign DNA, right? So restriction of enzymes that cut DNA at specific sites, leaving sticky ends that facilitate DNA recombinations, right? These two technologies are utilized in the we can say removing the unwanted DNA portion and inserting the wanted DNA portion. So here the latest technologies are there. CRISPR-Cas9 technology has come. So it is a revolutionary technology. It acts as a molecular scissors So it will cut the CRISPR-Cas9. This enzyme will cut the DNA portion which is unwanted, and it will introduce the wanted portion of genetic material. So with the arrival of this CRISPR-Cas9, we can say enzymes, the biotechnology or the gene therapy has become much more revolutionized. So try to remember this aspect also. Right. If we understand the tools and the steps in recombinant DNA technology, requirements are cell culture carrying the required gene, restriction, endonuclease enzyme for DNA cutting. So this is, many steps are involved in the we can say recombinant DNA technology. So for further knowledge, you can go through all these processes and steps, right? If you understand the significance of recombinant DNA technology, so it enables precise genetic manipulation for various applications in medicine, agriculture, industry, environmental science. So in agriculture, we can say genetically modified organisms, we can say, which are basically pest resistant, or they are resistant to climate variations like climate resistance or resilience. Right. Industry also, we can produce various enzymes, etc., drugs, enzymes, etc., at a mass scale. So environmental science also, they are useful. We have seen just now, bioremediation, etc. So it can be useful in these kind of processes. Right. Now we will understand the applications of genetic engineering. So in some detail, we will see the applications of genetic engineering. Right. First one is protein manufacture. Right. So bacteria and yeast, they have been used for production of cheese, alcohol, and antibiotics. This we have seen through the recombinant DNA technology, and even the gene cloning, they have led to the development of a new industry for manufacturing proteins. Right. Commercial success. So the production of human insulin in 1982, it marked the first commercial success for recombinant DNA technology. Similarly, we have been able to produce the therapeutic proteins, so which have we can say therapeutic values, right? So various proteins with the therapeutic values such as insulin, growth hormone, and cloning factors are now available in large quantities through the genetic engineering, right? If we see the enzyme production, so enzymes are produced from cloned genes offering specific industrial applications, right? So if we see the examples, proteases, they are used in manufacture of detergents and meat tenderizers. Similarly, amylases, they are utilized in production of beer, bread, and textiles. Next, glucoisomerases, they are used in making corn syrup, which is sweeter than sucrose and used to flavor soft drinks. So all these are possible through the enzyme production. Next one is enzymes are fragile and require specific handling, often being entrapped in gels or encapsulated in small artificial cells. So try to remember this property also. Similarly, biotechnology is used in production of antibiotics also. So genetic engineering research aims to manipulate biosynthetic pathways for the synthesis of antibiotics, right? So new antibiotics, they have been developed through genetic manipulation, contributing to advancement in human health. Similarly, production of vaccines has also been made possible through the biotechnology. So the bioengineered vaccines such as those for rabies and hepatitis B, they are developed by inserting a gene for the antigen protein into a plasmid, right? So bacteria containing recombinant DNA, they generate large quantities of antigen protein, which is then added to the vaccine. Similarly, the vaccines stimulates the production of antibodies against the antigen. Right. So these are the applications of biotechnology in various fields. Right. Now we will understand about transgene and transgenic. Right. So genetic engineering has enabled the production of organisms containing genes from other species. So gene from other species is introduced, then it is called as transgene. Right. So that particular organism, or we can say organ, whether it is plant or we can say microorganism, it is called as transgenic organism. Right. So the foreign gene introduced into an organism is termed as transgene, and the organism which is carrying this foreign gene, it is known as transgenic. Right. Right. If you see the usefulness of transgenic organisms, they help in providing improved yield. So the desirable traits can be introduced or enhanced in agricultural plants and domestic animals, particularly in cattle, to achieve better productivity. So transgenic animals, they, for example, if milk animals are there, they can produce more milk. Right. Right. Similarly, production of valuable products. So transgenic plants and animals can be engineered to produce valuable substances. For example, best example is the golden rice. Golden rice is there. So basically, golden rice has certain characteristics which can we can say which contains the micronutrients. So micronutrients, it contains micronutrients, and it prevents the people who are consuming golden rice from the micronutrients which are actually deficient, they are given to the human beings. Right. Right. So if we understand the methodology of production of transgenes, so there are basically two ways are there, two methods are there. First one is micro-injection method, and the second one is retroviral vector method. So these two methods are there. So they are, we do not go into the technologies, technical aspects. So if you are interested, go through the processes. I have provided the detailed description about these two, we can say, processes. The retroviral method and the micro-injection methods. So steps I have provided. If you are interested, you can go through the two processes. If we see the examples of transgenic plants, right, first one is better example, cotton. In India we call it as BT cotton. Right. So cotton, it is engineered to resist attacks by worms, reducing the need for chemical pesticides. So basically there are three, we can say, genetically modified, we can say, cotton varieties have been introduced till now. So those varieties are called as ball gourd 1, ball gourd 2, ball gourd 1, ball gourd 2 and ball gourd 3. Right. So basically it is to fight so whatever the ball is there before the fiber comes into open. So one particular pest attacks the ball and it eats away the ball, cotton ball. So there will be we can say a poisonous poisonous enzyme will be there within the plant of the cotton plant itself. So when that particular pest eats the part of the plant, it will die because of that, we can say, poisonous element. So it is made possible through the introduction of foreign gene. So basically it is called as BT cotton because Bacillus thuringiensis, a particular microorganism is there. So the enzyme acquired from this bacteria, Bacillus thuringiensis, it has been introduced into the we can say genetic material of the cotton plant. Right. So in this way the genetically modified cotton plants have been introduced in India. So there are many issues are associated with. So in the initial stages, the plant was successful. The we can say pest attack, which was eating the cotton balls, it has been reduced. For the problem is the particular whatever the pest is there, that particular pest, it also went into, we can say, genetic modification. It also undergone the genetic modification or genetic mutation, and this particular pest has acquired immunity for this whatever the, we can say, poisonous element was there. The particular pest has acquired through the genetic mutation, it acquired a resistance for the agent that has been introduced. So pest again started acquiring, started eating away the balls. So to overcome this, varieties ball gourd 2 has been introduced, and ball gourd 3 also has been introduced. So when we discuss the mains topics, we will try and go into some more detail about these aspects, how the particular pest it has the particular pest has been called as bollworm, right? It is known as bollworm, and the bollworm kept on obtaining the resistance for this whatever the genes have been introduced through the genetic mutation, right? So when we discuss the mains topic, when we discuss hybridization, and when we discuss the genetically modified organisms, there we will discuss in detail about the BT cotton. So remember this point also, it is the only it is the only crop, we can say, only crop or only product which is genetically modified product, GM product, that is allowed for commercial cultivation, or we can say cultivation freely in the we can say agriculture in India. So in India, it is the only BT genetically modified crop that is allowed for cultivation. So earlier there was a proposal to introduce BT brinjal also. So after many we can say public consultations, there was negative feedback from the people and also from the experts. So the introduction of BT brinjal has been we can say put on hold for a long period of time. It has been almost 10 years, right? Similarly, 5 to 6 years before, there was a proposal to introduce the genetically modified mustard also. So basically these two BT brinjal or BT cotton for that matter, these were the GMOs, genetically modified organisms, brought up by the foreign companies. But when it comes to the GM mustard, the Indian scientists, they have produced this genetically modified mustard. However, this also faced severe restriction, we can say severe resistance, and it also not introduced for commercial, we can say commercial cultivation. It has not been introduced until now. So we can say till now the only crop that is commercially allowed for commercial cultivation, only genetically modified crop is cotton, right? So there are also a lot of issues associated with BT cotton. When we study the mains topics, we will try and discuss in some more detail about this particular BT cotton, right? So examples are cotton is there, BT brinjal is there, BT mustard I have just now explained. Similarly, there are proposals for introducing the corn, BT corn and soybean also. So these are modified to be more tolerant to drought and pesticides, enhancing the crop resilience, right? If we understand the industrial and medical applications, so transgenic plants can also be utilized as factories to produce medically and commercially valuable proteins. For example, genetically modified potato and tobacco plants can yield serum albumin, which is used in preparations for patients with burn injuries and others requiring replacement of body fluids. So these are some of the applications of genetically modified plants, right? Next one is we understand about transgenic animals or genetically modified animals. So mice is there. As you all know, mice is used extensively for we can say whatever the we can say research of drugs or new medicines. First they are tested on the mice, then they will be tested on the human beings. So for testing of the drugs, the genetically modified mice, or we can say cloned mice, they are used for conducting these researches. So despite difficulty in generating transgenic animals due to animal cells reluctance to accept plasmids, transgenic mice are routinely produced in laboratories worldwide. So they are created through micro injection of foreign DNA into mouse eggs. So for example, gene for growth hormone from rats was micro injected into mouse eggs, resulting in mice that grew larger than their littermates due to the integration and expression of the rat gene in mouse DNA. So in this way, mice has also been we can say genetically engineered mouse has been produced. So another example is the rats and mouse. They are used in research. Research of the drugs. So genetically cloned mice or rats are used for the conducting the drug for doing the drug tests also, right? Similarly, goats have been produced, genetically modified goats. So transgenic goats, they have been developed by injecting a fertilized egg with recombinant DNA containing goat gene sequences spliced with human genes for tissue plasminogen activator TPA. So goat milk containing TPA has proven beneficial for heart attack and stroke patients by dissolving blood clots. So this is in this way goats have been useful. So whatever the milk consumed by the genetically modified whatever the milk produced by genetically modified goats, it has been beneficial for the patients of these patients, stroke patients and heart attack patients. Similarly, cattle. So transgenic cattle livestock have the potential to produce large quantities of drugs more rapidly and economically than bacteria, which require cultivation in large industrial vessels. So cattle, instead of using microorganisms, so we can use cattle for production of useful, we can say proteins or any other enzymes. So using cattle will become more easy than using microorganisms. Similarly, Chinese hamster. So genes for blood clotting factor will have been inserted into the Chinese hamster ovary cells, allowing for the production of this vital factor. So this factor is crucial for patients with hemophilia A and can now be generated through recombinant DNA technology in Chinese hamsters, eliminating the need to source it from human blood and reducing the risk of transmitting diseases such as AIDS. Right. So these are the benefits of transgenic animals and examples of transgenic animals. Similarly, bacteria engineered to be resistant to mercury can effectively process metallic mercury, which poses a significant risk to the nervous system, into non-toxic compounds. So by metabolizing mercury, these bacteria contribute to reducing the harmful effects of environment and impact on environment and human health. Similarly, earlier I have given the example of oil chopper also. So it also treats the oil pollution. So here this particular we can say bacteria, it is removing the mercury from the environment by degrading it, so that the harmful effects of mercury have been reduced on the human beings and also on the environment. So we can say this is also one technique of bioremediation. Right. Next, we understand gene therapy. So it holds a significant promise for treating genetic disorders that can lead to serious health complications and premature death. Right. This therapeutic approach, gene therapy, it involves replacement or manipulation of defective genes to correct the underlying genetic disorder. So just now I have explained the process of gene editing. So here it is the chromosome. So undesired part or defective part is removed. It is removed and a new proper functioning part, genetic part, is introduced. Right. So if we see the genetic definition of gene therapy, it is a technique where patients are provided with healthy genes to replace or enhance the functioning of defective genes inherited from their patients. Purpose of the gene therapy is, the primary goal of gene therapy is to replace or alter the defective genes, thereby addressing the root cause of the genetic disorders. Example, we have seen sickle cell anemia, diabetes mellitus. So all these are genetic disorders, we can say genetically, diseases that are coming genetically. So this root cause of genetic disorders can be addressed through the gene therapy. Right. So by introducing the functional or normal genes into the human cells, gene therapy aims to correct the inherited defects and restore normal cellular function. Right. Right. So if we see the implementation, in gene therapy, healthy genes are introduced into the genetic material of human cells, typically through vectors such as virus and nanoparticles. So here we have seen one example earlier. Plasmids are used as a vector. So apart from that, viruses can be used as a vector, or nanoparticles can also be used as vectors. So yesterday we were studying nanotechnology. So nanoparticles produced through artificial processes using nanotechnology, those particles can also be used as vectors. Right. These vectors, they deliver therapeutic genes to target cells where they integrate into cellular genome and begin producing the necessary proteins. Right. This is the process involved in gene therapy. Right. So if we understand the approaches to human gene therapy, so gene therapy is especially predominantly being used in human beings. I mean, it is, we can say we are in the initial stages. There are lot of apprehensions, and we can say we are in the initial stages of gene therapy, and there are several approaches for gene therapy. First approach is somatic gene therapy. So somatic gene therapy, it targets non-reproductive cells. So reproductive cells, you should know, they are egg and, we can say, right. So basically the somatic gene therapy, it utilizes non-reproductive cells. So reproductive cells are also there. So basically reproductive cells are egg and sperm. These are reproductive cells, right? So somatic gene therapy, it uses non-reproductive cells. So cell of the skin, cell of the liver, we can say. So when these cells are used for gene therapy, it is known as somatic gene therapy, right? So it uses non-reproductive cells for gene editing to correct genetic defects confined to specific organs or tissues. So for example, if I have a problem in liver or heart, so cells from that particular organ will be used. Cells from heart or cells from the liver. Then it is known as somatic gene therapy. So here different methodologies are there. One is ex vivo gene therapy and another one is in vivo gene therapy. So we need not go into this much detail. So if you are willing to know more about these technological processes, you can study the aspects mentioned below. So basically two procedures involved are ex vivo gene therapy and in vivo gene therapy. Right. Another approach is there. Second approach, it is germline gene therapy. So this approach involves genetic modification of germinal epithelium cells or zygotes to introduce remedial genes into the we can say future generation. So zygote is generally egg cell and a sperm cell. When these two are meet, the zygote will be formed. Zygote will be formed. We can say it is a zygote is a fertilized egg, right? So when modifications are made in the fertilized egg, it is known as germline gene therapy. So currently research on human gene therapy primarily focus on correcting gene defects in somatic cells as opposed to germline cells. So we do not reach here. Generally present the focus is on the somatic gene therapy only. I mean, it is correcting the defects at the individual organ levels only, right? Next one is third approach in genetic, we can say gene therapy is antisense gene therapy. So definition, if we see antisense gene therapy, it is designated to reduce or prevent the expression of specific genes by introducing nucleic acid sequences complementary to the mRNA of the target gene, right? Methodology: an expressible gene is cloned in reverse orientation, producing antisense RNA that pairs with the mRNA, preventing translations, right? If you see the applications, antisense therapy holds promise for treating human genetic disorders and cancers where overproduction of a gene product disrupts normal cellular function. So cancer, how cancer happens? The overproduction of cells. So it is because of the cell reproducting function of a particular gene. So through antisense therapy, that particular gene which is producing the cells, that particular property of that gene will be silenced through the genetic engineering. So in that way, cancer will be cured, right? So it has been explored for treating malignant glioma and has been resulted in the development of tomatoes with extended shelf life. So it is, the process has been successful in tomatoes also. So now through this property, tomatoes with more and more shelf life are being produced, right? Right. So current gene therapy, as I have told also, there are many concerns also. We are at present at the initial stage of the gene therapy, and already there are concerns taking about cloning. So some concerns are there regarding cloning also. So there are many ethical issues are also there. So how far gene therapy can be used? So when it comes to cloning, so whether a particular replica like me can be produced or not. So what are the consequences if a exact replica of mine will be produced? So what are the consequences of it? Is it ethical, etc.? So if we see other ethical aspects, like it is okay to treat the ailments or problems. So if I have a particular disease, so it is okay to treat that particular disease by genetic engineering, gene therapy. What if a particular person, he is trying to enhance his capabilities, like he want to look more handsome, or a particular person she want to look more beautiful. So if there are we can say that is being done through the we can say gene therapy, is it allowed? Will we have to allow it or not? So these kinds of ethical aspects are there. For example, a person wants to increase his lifetime by we can say gene therapy, whether we should allow it or not. So similarly, another concern is ethical concern is, so in this process, the eggs, reproductive organs or reproductive items like eggs and sperm cells are used. So in this process they are utilized. So some we can say some norms or societal norms are there which oppose tinkering with the reproductive elements like eggs and sperm. So all these are many ethical concerns are there. So there is no agreement on that. So these are some of the aspects associated with the ethical aspects of gene therapy, right? Right. So these aspects whatever I have discussed, so they are also useful in the mains perspective. I mean in the ethics also we can expect a question about gene therapy, etc., etc., because it is an evolving technology and there are many concerns from various societies also. So try to focus on this aspect, right? So current applications of gene therapy, if we see, so currently it is used to treat diseases such as AIDS, hemophilia, leukemia, lung cancer, SCID. So the somatic gene therapy is somatic cell gene therapy is used in treating all these diseases. So for this it is acceptable, till now it is acceptable to treat the diseases. It is okay. So what happens if a particular person is using them to enhance his capabilities? To, I mean, example just now I have quoted, like a person wants to increase his age. So what about that case? Whether whether it has to be allowed or not, right? Some other limitations on gene therapy are restriction to somatic cells. So it is confined, present it is acceptable to only non-reproductive cells. I mean, use of reproductive cells like eggs and sperm cells are, it is not yet allowed, right? So now gene therapy is allowed to limit to somatic cells, meaning that genetic improvements made in treated individuals cannot be passed on to their offspring. So whatever the modifications are there, they have now been confined to the individual only. They cannot be passed to the offspring, right? Potential for unintended genetic effects. So there is a risk of random integration of DNA into human chromosomes, potentially leading to the inactivation or activation of normal genes. This could result in enzyme deficiencies or uncontrolled cell division, leading to cancerous growth. So this threat is also there. Similarly, stringent safety standards, right? So producers of gene therapy must adhere to strict safety standards established through animal trials to minimize risk to patients, right? Similarly, limitation to single gene disorders. So gene therapy is currently most effective for treating diseases caused by defects in a single gene. The normal gene must be identified, cloned and made available for transplantation, right? Right. So concerns, if we see the concerns regarding the germline gene therapy. So germline gene therapy, it involves manipulation of sex cells, the egg and sperm. So it is not currently practiced due to concerns about introducing unforeseen genetic changes in the future generations. So it has been not allowed now because we don't know the consequences. What are its we can say long-term consequences. So because of this reason, the gene therapy, it is now not allowed in the sex cells, right? So this approach is highly controversial and raises ethical, safety and regulatory concerns. So just now I have discussed the examples. So because of this approach raises many concerns, including ethical concerns, safety concerns and even the regulatory concerns, right? Right. So this is some information about biotechnology, and we have also discussed extensively on the we can say genetically modified organisms and also the gene therapy, right? Similarly, there are some examples about plants. We have discussed genetically modified organisms, genetically modified crops like cotton, and I have also explained the controversies associated with the genetically modified crops. I have given the example of we can say this one, BT cotton. So there are concerns about the BT cotton because the pest ball gourd, it gained we can say it gained the resistance for particular whatever the genes that have been introduced in the plant. Other concerns are there, like they are very costly. The genetically modified, we can say, seeds, they are really costly. So through the genetically modified we can say crops, the seed industry has been commercialized, or we can say corporatization of agriculture, agricultural. So basically these plant varieties have been brought by the multinational companies, not even the national companies. So they are multinational companies are bringing these genetically modified seeds. So there are a lot of apprehensions, not only about the cost but also about the future accessibility of these seeds by the farmers, right? Similarly, there is a concept called terminator genes. So there is a concern about the terminator genes. So through the modification of genes, the seeds can be made like that they will give only one crop. So after, if a farmer tries to use the seeds coming from that crop, the seeds will not be fertile. So again the farmer has to purchase new seeds from the company. So in this way there is a huge threat to the farming community also. So I mean, there are many aspects about the genetically modified crops. So when we discuss the mains aspects, we will go deep and also discuss these aspects about the all these aspects about the entry of multinational companies and the use of terminator genes, what will happen to the agriculture, and what about the interest of the farmers. We will try to discuss these aspects also, right? Now we will see some of the questions that have been asked in the examination from this topic, biotechnology. First question, it is asked in 2021. The question is, with reference to the recent developments regarding recombinant vector vaccines, consider the following statements. Statement 1 is, genetic engineering is applied in the development of these vaccines. Yes, this statement is correct. Bacteria and viruses are used as vectors. Yes, we have understood bacteria and viruses and also plasmids, they will be used as vectors. So both the statements are correct. Correct option is option C. Both statement 1 and 2 are correct. Next question, it is asked in 2021. The question is, ball gourd 1 and ball gourd 2 technologies are mentioned in the context of option A, cloned propagation of croplands. Option B, developing genetically modified croplands. C, production of plant growth substances. D, production of biofertilizers. So just now we have understood ball gourd 1 and ball gourd 2, they are the BT cotton varieties. Similarly, ball gourd 3 is also there. So we have understood. So these genetically modified cotton crop has been developed to resist the bollworm, which is eating the cotton balls, right? So correct option is option B, developing genetically modified croplands, especially the cotton, genetically modified cotton, right? Next question, it is asked in 2019. Question is, what is Cas9 protein that is often mentioned in news, right? Option A, a molecule scissors used in targeted gene editing. Option B, a biosensor used in the accurate detection of pathogens in patients. C, a gene that makes plants pest resistant. D, a herbicidal substance synthesized in the genetically modified crops. So all the options are closer, relevant to the topic of biotechnology, but the correct option is, as I have already explained, so CRISPR-Cas9 technology is there. So in that, Cas9 is the name of a protein which acts as a molecular scissors, which cuts the undesired part of a, we can say, genetic material and introduces the desired genetic material. So it is a molecular scissors used in targeted gene editing, right? So these are some of the questions asked. So when we were studying the nanotechnology also, I think artificial intelligence, there was in the options there was mention about genetic engineering, I guess. Like engineering, or especially the question was of CRISPR-Cas9. CRISPR-Cas9, right. So this particular option was also there in the pairing. The question we were studying, we were seeing the question about the artificial intelligence. There one option is there about CRISPR-Cas9 technology, right. So these are some of the questions that are asked previously in the examinations. So this is very important topic from the point of view of examinations. Please focus on the science and technology part also. So yearly there are 10 questions, at least 10 questions are being asked from the technology, science and technology part, right. So this is all for today. Thank you for joining the lecture. See you next time. Until then, have a good day. Bye.